Where's the... So our next speaker is Timothy Durham from the University of Washington and from Bill Noble's lab, and he'll be talking about epigenomics data imputation. It's Durham, this one.
I see, okay.
1 minute, 11 minutes.
Okay, thanks. Okay, good morning, everyone, and I'd like to thank the organizers for giving me the opportunity to tell you about my work today. I don't think I have to convince anyone in this audience that a central question in the field is how diverse cell types can interpret the genome to selectively express the genes that are relevant to their function in the body. And we know that this is accomplished through the concerted action of many different protein and RNA species and even the 3D conformation of DNA in the nucleus. The way we usually assess these different epigenomic factors is by performing a panel of assays across our different cell types of interest. Each of these assays makes measurements genome-wide. One of the limitations of this approach is that these assays are still time-intensive and relatively expensive to do, and so there are relatively few cell types that actually have complete coverage of these epigenomics assays. We can visualize this with a subset of the Roadmap Epigenomics Consortium data. So here I'm showing this as a 2D matrix with 127 cell types and 24 assays. These assays include histone modification ChIP-seq data sets and chromatin accessibility assessed with DNA's hypersensitivity sequencing. And what you'll notice is that there are of 3,048 possible experiments. We have 1,014 that have actually been performed in the lab. So 66% of the data are missing just in this subset. And if you expand this matrix to the whole world of different cell types and epigenomics assays that you could measure, the missingness is quite a bit more than that. The other thing to note about the structure of this problem is that implicitly this is actually representing a 3-dimensional tensor because each of these is a genome-wide assay. So we have this problem where we're trying to learn about these missing data entries in this large, long, skinny tensor. So for anyone who has a cell type that they're interested in, but these assays haven't been performed yet, we'd like to be able to say something about what the other assays would show. This is not a new problem. Back in 2015, Jason Ernst and Manolis Kellis published a method, chromimpute, that actually performs quite well. And so their strategy, briefly, was to train an ensemble of regression trees for the data from the same assay but different cell types, and the same cell type but different assays, and impute based on that data. And this raised the question for us of whether we could train a single model that would jointly learn from all of the data in this tensor at once and impute the missing data all at once. And the technique that we chose was tensor factorization. For those familiar with matrix factorization, this is a kind of a generalization of that approach to a third dimension. And in this approach, we try to factorize or decompose this tensor into 3 low-rank matrix factors that share this dimension k. And each of these matrices captures patterns along each of the dimensions in the tensor. So we have one for cell types, one for assays, and one for genomic loci. And you can reconstruct this tensor after training these factors by taking the outer product of corresponding latent factor vectors and summing the resulting tensors by element-wise. This is the objective function that we optimize. So we have 70% of these experiments we designate as training experiments, and we minimize the squared error of our imputed values to these training experiments, and regularize these matrices with L2 regularization to prevent overfitting. And we train this model using parallelized stochastic gradient descent with the atom optimizer. And we parallelize this tensor data structure across an Apache Spark cluster. We use a subset of the genomic positions to train these cell type and assay factors, which are shared across the genome, and then fix those factors and sweep across the genome using a second-order update to make our final genome parameter updates. The other thing I should note is that we found that this works best with an ensemble prediction. So we trained 8 models like this, and the data that I'm going to show you are the average of the imputed results from those 8 models. So this is these are 4 different marks, 4 different assays from that we imputed, paired with their observed data. And what you'll notice is that we capture the shape of the signal quite well, although I'll note that we tend to impute a lower amplitude of the signal than the actual observed signal. So on a more global scale, how does this compare against other models? We compare against 2, one baseline model and Chrome impute. And so the baseline is a main effects model in which each genomic position is treated independently. And we impute any value in this matrix by taking the sum of the global mean at that position of the training data, the row residuals, and then the average of the assay residuals, which are on the columns. And predicted outperforms this baseline model on 85% of the assays in the roadmap, this roadmap subset. And if we add in the Chrome impute, what I'm showing is the relative performance of these 3 models on each of the different experiments in our compendium. And I've highlighted the regions of this ternary plot or 2 simplex with the model that performs best in that region. And so you'll see in red, predicted outperforms the other 2 models by this mean squared error metric on 62% of the experiments. This doesn't hold necessarily depending upon what quality measure you use. So if you use the genome-wide correlation, Pearson correlation, Chrome impute outperforms predicted, but if you'll notice that these experiments are right along the boundary here between the predicted and Chrome impute models. And it turns out that if you average the results of predicted and Chrome impute, you get the best model. So then the average model performs best on both of these quality measures. So we think it's doing a good job of producing imputed data. Now, how might you actually make use of this? We think one of the most useful cases for data imputation, as I mentioned earlier, is that if you have a cell type that is undercharacterized. And to simulate this situation, we took one cell type, the CD3 primary cells from cord blood, and trained models on holding out all of the data except for 1 or 2 data sets for this cell type, and compared how the model performed. And so these blue bars represent the performance on all of the training data with all the training data available. If we only train on H3K for ME3, an active promoter histone modification, we see that the performance is actually pretty similar. So we think the model can effectively take information from other cell types and assays and apply it to new ones. This doesn't hold for every assay. So if you try a different assay like histone 3 lysine 4 monomethylation, you don't get this quite as good performance. And you can take assays that have more or less orthogonal information, like a H3K for ME3 and K9 ME3, which the latter marks repressed regions, and make improvements on some of these in some of these cases. So assuming you have a cell type of interest that you've imputed the data for, is it actually useful? Can it tell you something about the biology of that cell type you're interested in? To address this question, we looked at non-coding human accelerated regions, which are regions of the genome that have more mutations in the human lineage than would be expected given their level of conservation across phylogeny. And it's thought that these are enriched for enhancer regions that are specific in the human lineage. And so we were able to cluster, we did a bi-clustering of enhancer-associated imputed signal across all of our cell types on these non-coding human accelerated regions. And we find that for 601 non-coding human accelerated regions, we can actually find imputed putative enhancer activity based on these marks, and that they cluster based on cell type. So we have this cluster of non-coding human accelerated regions that would have putative enhancer activity in brain and embryonic stem cell cell types. To assess whether this actually makes sense, we compared using the adjusted RAND index to clustering, doing the same clustering with just observed data, and we find that the clustering results are highly similar. We also compared against the results from Enhancer Finder, which was published in 2013 and used an ENCODE cell line data set to do a similar, similar clustering, and we find that we're very similar to those results as well. Compared to two negative controls. So if you shuffle the positions of the non-coding human accelerated regions, you lose this similarity in clustering. And if you cluster based on non-enhancer associated imputed marks, so K27ME3, K36ME3, and K4ME3, you also lose this clustering. So with that, I'd like to thank you for your attention and thank all my co-authors and UW IT Cloud Computing for helping with computational support, my committee, funding sources, and in particular ISCB for a travel award to come talk to you today. And if you're interested in learning more or trying the data, training your own model or looking at the imputed data that we produced, you can access that through the ENCODE portal. The code is on our Bitbucket website along with tutorials for how to set this up on Amazon Web Services to try it on your cell type of interest. And this was published earlier this year. Thank you very much.
The next speaker, Akshay Balasubramanian, please come to the podium. Thank you.
Very nice talk. I had a question sort of with regards to the representation of the signal and then the evaluation metric of mean squared error.
Oh,
so
sure.
my understanding is you've been using sort of the negative log base 10 p-value. That's what Chrome Impute was done on.
That's right,
and then,
yes.
I mean, you would expect when you're sort of have higher quality data, like a greater effective sequencing depth, that those values would tend upwards. So then the mean squared error might not be the best evaluation metric in that situation. So can you comment on that?
Yeah, so that's a good point. We did use, the data sets that we used are the consolidated data sets from Roadmap, which were downsampled to the same sequencing depth. So we think they are comparable. We also transformed the data using an inverse hyperbolic sine transform to stabilize the variance and kind of bring the error model closer to Gaussian.
Yeah, but I mean, the question is, when you then impute it, you would potentially get a higher quality data set, which could get better negative log base 10 p-values or higher on average than the original one. We can talk.
I'm not sure I totally understand. That'd be good to talk.
So if I understood your results correctly, you seem to get performance almost as good when you train on H2K4ME3 than when you train on everything. And I was wondering if you've looked at where your model does well and where it does badly. Does it tend to do better when you're closer to a transcription start site?
Yes, we've done a little bit of that. So the model definitely has a harder time at places where there's more cell type specific activity. If we kind of partition the genome based by peaks and look at peaks that are accessible or found in all cell types versus lower subsets of cell types, you do see kind of lower performance in those lower ones. But we find that we actually have a pretty good precision for the peaks that are present in low, kind of fewer cell types. So these cell type specific peaks, we don't always have the very high recall, but the peaks that we do call are there.
Great, thanks.
Yeah.
So are you imputing p-values or are you imputing the actual count? Because in the tracks you showed that you are actually showing the counts, but the tracks on ENCODE seem like it's p-values.
Oh, sorry if I gave that impression. These are not counts. We are imputing the negative log 10 p-value. tracks. We're imputing based on those, so these are imputed minus log 10 p-values. It's on that scale.
All right, let's thank Tim once again.
Thank you very much.